Every molecular biologist asks the same question: how much DNA do I really have? The answer can make or break your PCR, sequencing, or cloning experiment. In this video, we will decode the science behind NanoDrop and Qubit, two powerful tools that measure your nucleic acid, and reveal when the speed matters more than accuracy and when precision can save your experiment. Let's demystify molecular biology together. Accurate quantification of DNA and RNA is the foundation of successful molecular biology. If you add too little template to your reaction, you risk failed amplification and wasted time troubleshooting. If you add too much, you can overload your reaction, create artifacts, and end up with unreliable data. The precision of your quantification directly impacts your experimental success. Historically, researchers estimated DNA amounts visually by running samples on an agarose gel and comparing band density to a standard ladder. This method was slow, subjective, and prone to human error. Fortunately, modern molecular biology has advanced far beyond that. Two technologies, the Nanodrop Spectrophotometer and the Qubit fluorometer, now make nucleic acid quantification fast, accurate, and reproducible. Let's start with the Nanodrop, a compact spectrophotometer that works on the principle of Beer-Lambert's law. The fundamental relationship that states light absorbance is directly proportional to the concentration of molecules in solution. Nucleic acids absorb ultraviolet light most strongly at 260 nanometers, thanks to the aromatic ring structures in the nitrogenous base. By measuring this absorbance, NanoDrop calculates the concentration of DNA or RNA in your sample. The beauty of NanoDrop lies in its simplicity and efficiency. You only need 1 or 2 microliters of sample, and in just seconds you get not only the concentration but also purity ratios that reveal possible contaminants. Here is how it works. Step 1. Blank the instrument using your elution buffer. Step 2. Load 1 to 2 microliter of your DNA or RNA sample. Step 3. Close the arm and measure. Step 4. Review your concentration and purity ratio. NanoDrop provides two key purity ratios, A260 by 280, which tells you about protein contamination. Pure DNA should be around 1.8 and pure RNA should be around 2. A260 by 230, which detects contamination from salts, phenol, or organic compounds. A pure sample typically reads between 2 and 2.2. This ratio helps you instantly assess the quality of your extraction before moving to sensitive downstream applications. The NanoDrop's advantages are clear: speed, simplicity, and no consumables. It's ideal for quick screening of multiple samples. However, it has one major limitation. It measures everything that absorbs at 260 nanometers. That means degraded nucleic acids, proteins, or even residual phenol can inflate the absorbance and overestimate concentration. NanoDrop cannot distinguish between target DNA or RNA and contaminants. Now let's look at the Qubit fluorometer, which uses an entirely different approach: fluorescence-based quantification. Instead of measuring light absorbance, Qubit uses special fluorescent dyes that bind only to DNA or RNA. These dyes emit light only when bound to their specific target, eliminating background interferences from proteins, salts, or other UV absorbing contaminants. Depending on the assay you select, for double stranded DNA, single stranded DNA, or RNA, the Qubit dye binds specifically to that target and emits fluorescence in the direct proportion to its concentration. Because of this selective binding, Qubit provides exceptionally accurate readings, typically within plus or minus 5% error, even for very low concentration samples that NanoDrop might miss. It can detect nucleic acid as low as 10 picograms per microliter, making it gold standard for next generation sequencing, library preparation, and other applications where precision is critical. However, Qubit does take a bit more time and effort. You will need to prepare the reagents, incubate samples, and use consumables assay tubes. Also, Qubit does not measure purity, it only gives you the concentration. Here is how a typical Qubit workflow looks like. Step 1. Prepare your standards using the kit's provided reagents. Step 2. Mix your DNA or RNA sample with Qubit's working solution. Step 3. Incubate for 2 to 5 minutes in the dark. And step 4. Measure fluorescence. The instrument calculates concentration using your calibration curve. Because of this selective binding, Qubit provides exceptionally accurate readings, typically within plus or minus 5% error, even for very low concentration samples that NanoDrop can miss. It can detect nucleic acids as low as 10 picograms per microliter, which makes it ideal for next generation sequencing library preparation. So, which one should you use? The answer depends on your workflow. Use NanoDrop for quick routine checks when you want to assess purity or screen multiple samples rapidly. Use Qubit when accuracy matters the most, before sequencing, cloning, or sensitive downstream experiments. In fact, the best practice is to use both. First, screen your samples with NanoDrop for purity, then verify concentration with Qubit before proceeding. This combined approach ensures both speed and reliability. Now, even the best instruments can give inaccurate results if not used properly. Here are some best practices to ensure consistent and reliable readings. NanoDrop best practices. Clean the pedestal between samples. Always match your blank with your buffer. Avoid air bubbles when pipetting. Monitor your purity ratios carefully. A260 by 280 less than 1.6 indicates protein contamination. A260 by 230 less than 2 indicates salt or phenol carryover. Qubit best practices. Mix dye working solution thoroughly to ensure uniform dye distribution. Verify that reagents are within expiry and stored at 2 to 8, protected from light. Use the correct kit. DNA high sensitive, broad range, RNA high sensitive, depending on your expected concentration. Always run duplicates to catch pipetting or reagent error. If duplicates differ by more than 10%, recheck calibration or pipetting accuracy. Paying attention to these small details ensures your data is both accurate and reproducible. The introduction of NanoDrop and Qubit transformed how laboratories approach nucleic acid quantification. They brought consistency and standardization to workflows across the globe. In NGS library preparation, Qubit ensures libraries are pooled precisely for efficient sequencing. In clinical diagnostics, both instruments help maintain accuracy in nucleic acid based tests from infectious disease like COVID-19 to oncology panels. And in high throughput research, automation systems now integrate both NanoDrop and Qubit for processing thousands of samples daily. Beyond accuracy and speed, these technologies democratized molecular biology, allowing even small labs to perform high quality quantification with minimal training. Let's summarize what we have learned. Speed versus accuracy. NanoDrop is fast and gives purity data. Qubit is slower but more accurate. Complementary tools. Use NanoDrop for initial QC, Qubit for final quantification. Workflow integration. Combining both improves reproducibility and reliability. Now let's take a small quiz for you. What does A260 by 280 ratio of 1.4 indicates? It indicates protein contamination. Why does Qubit often give lower concentration than NanoDrop? Because Qubit measures only intact DNA or RNA, not contaminants. When would you choose NanoDrop? For rapid screening, purity checks, or when reagent cost is a concern. Accurate nucleic acid quantification may seem routine, but it's one of the most crucial steps in any molecular biology experiment. The NanoDrop gives you speed and purity insights, the Qubit gives you precision and specificity. Together, they empower reliable and reproducible science. In the next episode of Molecular Biology Demystified, we will explore restriction enzyme and DNA ligation, the molecular scissors and glue that built modern genetic engineering. Subscribe, share your experience in the comments, and keep exploring the science that powers discovery. Thank you for listening.